My laboratory is focused on determining the molecular mechanisms that underlie drug addiction. Specifically, we're interested in cocaine addiction. We know that when you use drugs such as cocaine, there are changes that occur in the brain, so in brain cells, and now we're actually focusing in the brain cell nuclei where genes are expressed, because using drugs of addiction changes the way that the genes are expressed. So we want to know how cocaine actually does this, because changes in gene expression underlie all the behavioral changes that we see in the brain. So we think that that's an important mechanism. And also, if we can decipher what these changes are or identify the targets that are involved in regulating cocaine addiction, then perhaps we can find novel therapeutics because they'll be specific to cocaine addiction. So what we like to do is to get down into the nucleus and actually look at these molecular regulators. I think it would be a good way to call them, or we would call them epigenetic marks. Now, these epigenetic marks can be inherited, so from parent to child. So if your mother's marks are different, she can pass that on to the child. And then also, what's changed in the mother or the father can be then passed on to the grandchildren as well. And we find this very intriguing. We want to see that what is the molecular changes in response to cocaine, describe that in the parent, find these targets, and then look at the offspring and see whether or not these have been inherited or not. We have two models. So we have a mouse model, which is a maternal transmission. So in this mouse model, the mothers get the cocaine, and then we follow the offspring. So we look at the children, the grandchildren, as well as the great-grandchildren. And then we have another model, which is a rat model, and this is a cocaine self-administration rat model, where these animals will actually press a lever to receive cocaine. So this model is actually a better description of drug abuse and human condition, because they voluntarily press the lever to receive the drug. We find that in both models, we see similar changes in brain activity, similar changes in protein levels, and it's in particular, we find changes in a protein called brain-derived neurotrophic factor. The molecular mechanisms or the regulation of BDNF expression is changed similarly in both models, and that this is a heritable phenotype. So far we've looked at the children and the grandchildren of both models, and they show similar changes. So we're very excited about this because we think that this must be something important, that we're seeing it in two separate models of addiction.